So the next sort of a block is about the regulation of the urine concentration. And before we go into this topic, I want to make it clear why we actually care about this. One of the main functions of kidneys is to conserve what? Water. So by regulating the urine concentration, we can essentially conserve water. When kidneys make urine more concentrated, they save water. When urine becomes less and less concentrated, they get rid of water. Remember that throughout our conversation, okay? Now, before we start, so this function of regulating the urine concentration is carried out by juxtaglomerular nephrons and juxtaglomerular nephrons only, not cortical, but juxtaglomerular. How do we measure the concentration of urine? You can imagine that urine contains multiple chemicals, from urea to potassium, to drugs, to glucose, amino acids, anything that gets filtered. Now, instead of trying to express the concentration of urine as, some kind of concentration of each individual component, we use a concept of osmolality. Osmolality is the number of solute particles, and here I have to correct myself, in 1 liter of water. Does that make sense? So it is measured in application to the renal system in the units called milliosmoles. So for instance, let me ask you this question to quickly understand if we get a grasp on it. Imagine that you have I'm going to explain it. So 1 mole of sodium chloride is 1,000 millimoles. Are you with me? If you dilute 1 mole of sodium chloride in 1 liter of water, you're going to have 1,000 millimoles of sodium and 1,000 millimoles of chloride ions. Are you with me? Okay, so this is 1,000, this is 1,000, and this is 1,000. So the osmolality of such solution is 2,000 milliosmoles. Now you can mix sodium chloride, potassium sulfate, and I don't know, calcium nitrate, glucose, urea, whatever, anything you want. Osmolality measures simply the number of solute particles without referring to specific particles. Are we clear? So normal osmolality of plasma is 300 milliosmoles. That includes all the sugar, salt, blah, blah, blah. Does that make sense? So the function of juxtaglomerular nephrons is to maintain the osmolality of plasma at 300 milliosmoles. And that maintenance is carried out by so-called counter-current mechanism. Counter-current refers to the process in which fluids flow in the opposite direction. Okay, so here the fluid goes from cortex to medulla, and here the fluid goes from the medulla back into the cortex. Does that make sense? That's counter-current mechanism. Do you understand what I'm saying? So the nephron loop, this yellow part, is the countercurrent multiplier. Okay? The function of countercurrent multiplier is to regulate the concentration of the filtrate. The countercurrent exchanger is Vasa recta, the capillaries that surround the nephron loop of juxtaglomerular nephron. Their function is to collect reabsorbed water and reabsorbed salts and carry it away from the medulla. Does that make sense? This system, countercurrent mechanism system, allows kidneys to maintain what is called the osmotic gradient from cortex to medulla. If you would look at the schematics at the bottom, you will notice that osmolality of extracellular fluid in the cortex and at the border between cortex and medulla, right here, is 300 milliosmoles. However, osmolality progressively increases as we go deep into the medulla, all the way to 1200 milliosmoles. Without trying to imply anything, any additional information, so far, you're keeping up, right? So, the most important take-home message so far is that you have two parallel systems, multiplier and exchanger. Multiplier refers to filtrate in the nephron loop, exchanger to the blood in the vasa recta. And also, the concentration of extracellular fluid becomes higher and higher as we move deep into the medulla.
So, with the countercurrent one, you said they collect the reabsorbed water and what?
So, countercurrent exchanger is vasa recta, the capillaries, and they collect reabsorbed water and salts.
So they deal with the blood.
Okay, countercurrent exchanger is vasa recta. Those are capillaries. There's blood in them. The function of that blood is to collect and take away reabsorbed water and reabsorbed salts. That makes sense? Now let's take a look at the countercurrent multiplier. Here's what happens in the nephron loop of juxtamedullary nephron. Let's trace the fate of the filtrate all the way from the glomerulus. So this here is the schematic representation of the glomerulus, the capillaries, and as we mentioned before, the osmolality of the plasma is 300 milliosmoles. Does that make sense to you? 300. So remember I told you that virtually everything from plasma except proteins is filtered into the renal capsule. So what's going to be the initial osmolality of the filtrate? 300, because everything is filtered. It's the same solution, right? So here's your 300 milliosmoles filtrate that, after going through the proximal convoluted tubule, enters the nephron loop. Now look what happens in what's called descending limb of the nephron loop. In this part, water is reabsorbed from the filtrate into the medulla of the kidneys. Remind me, please, how do we call the process in which water moves towards the area with the higher concentrations of solids? That's osmosis. So in the nephron, in the descending limb of the nephron loop, water is reabsorbed via osmosis. So the numbers on the left represent the osmolality of interstitial fluid. So that's the osmolality in the medullary tissue. Does that make sense? So when filtrate with, say, osmolality 300 gets to about this area, filtrate has osmolality of 300, medulla has 400, you have osmosis. Does that make sense? Do you understand what I'm trying to say? Do some like head movements or anything.
Can you say it again?
So, look at this. Filtrate has the osmolality of 300. Let me erase some stuff here. So it has osmolality of 300. When this 300 milliosmol filtrate reaches that area right here, its concentration is lower than the concentration, that osmolality of interstitial fluid. Does that make sense? So water will start moving into the interstitial fluid, into the medullary tissue. Water will be reabsorbed by osmosis. Right? Now, you have to understand, it's not the process that is done in steps. Like water comes into the 400 region, sits there and waits, or, goes into the medulla while the filtrate patiently waits for all water to be reabsorbed. It's the continuous process that happens all the way through the descending limb. At every millimeter, or should I say micrometer, of the descending limb, the osmolality of the filtrate gets equilibrated with the osmolality of the medulla. Does that make sense? At every micrometer of the descending limb, the osmolality of the filtrates gets equilibrated with the osmolality of the medulla by water reabsorption. Water constantly leaves the filtrate and goes back into the medulla. We good? Now, what's the concentration at the very bottom of the nephron loop? It's very high, it's 1200, right? Make sense? So, at this point, filtrate has the highest concentration. What's the osmolality? Sorry, what's the osmolality of the filtrate at the bottom? 1200. This is why this loop is called countercurrent multiplier. It multiplies the concentration. So far, up until this bottom point. Does that make sense? Now I'm going to ask you a very, very important question, like really important question. So the volume of filtrate that enters the nephron loop, I'm going to call it V1. It's right here. And the volume of filtrate at the bottom of the nephron loop, I'm going to call it V2. Listen carefully. Think. It's actually a simple question. Which volume is bigger? V1. Do you understand why? What happens in the descending limb? Water gets reabsorbed. So water leaves the filtrate, right? So the volume of filtrate progressively decreases. Remember, that's a schematic. Like, don't pay too much attention to to the actual diameters and stuff like that. You see what I'm saying? So the volume of the filtrate decreases because water gets reabsorbed. Is that understood? Got it? Now, what happens in the ascending limb? In the ascending limb, sodium chloride gets reabsorbed, either through the pumps or at some point a little bit through the facilitated diffusion. The point that I want you to make here is that in the ascending limb, there's no water reabsorption. Water stays in the filtrate. What gets reabsorbed is the sodium chloride. Are you with me? As sodium chloride leaves the filtrate, what happens to the osmolality of the filtrate as it moves up through the ascending limb? Decreases. Is that understood? We take the salt away. Now, what about the volume? That is a hard question. The volume V3 compared to V2. Say again? It still decreases, not that dramatically, very marginally, but it will. Because you're still taking the stuff out. Okay? Now, before we move any further, I want to kind of reiterate what we said. Filtrate enters the descending limb of the nephron loop, and while it moves down, deep into the medulla, only water gets reabsorbed. I want to stress it for you. Descending limb is permeable only for water. Nothing else can go through, just water. We clear? The filtrate reaches its highest concentration at the bottom of the loop, and then starts to move up, and the salts get reabsorbed, mainly sodium chloride. So ascending limb is permeable only for sodium chloride. Water doesn't go anywhere. We clear? Quite paradoxically. Oh yeah, last question. Compare V1 and V3. Which one's bigger? Which one's smaller? V1 is bigger, much bigger. So now let's put it in the words. When filtrate goes through the nephron loop of juxtaglomerular nephron, its volume decreases. Does that make sense? Because water and salts leave the filtrate, so the volume of the filtrate goes down. Are we clear? So by the time we end up at this point, we have less filtrate than we had in the beginning because we conserved some water. Does that make sense? Now look at the concentration. It is now not 300, but 100 milliosmoles. Can you see that? And from now on, so far, the entire proximal, the entire distal convoluted tubule, all the way here, the concentration of filtrate is 100. So think of it like, I'm going to put it kind of in a perspective for you. So you have a filtrate that enters the nephron loop. When it comes out of nephron loop, its volume is much lower, but it's diluted. You see what I'm trying to say? Compared to its original concentration, it's diluted because both water and salts are leaving it. That makes sense? Now, one really, really important statement. And that's just, take it for face value. That reabsorption of water here, it's not regulated. Okay? It's called obligatory water reabsorption, or obligate. Do you understand the word obligate? It must happen. No matter if you are overhydrated, dehydrated, normally hydrated, that water will be reabsorbed. Does that make sense? So this water reabsorption doesn't really regulate the urine concentration. It helps you to conserve water, but there is no regulation involved. Any questions before we move on?
Yeah.
Sure.
With the V1 and V3, so the volume is still low like it was in V2.
It's even lower than V2.
It's even, okay, so it's just really diluted.
It's paradoxical, but it's diluted because a lot of salt is taken out as well. Does that make sense? Think of it, I know that concentration is not American chemistry education forte, but if you think about it in the terms of, say, a crowd of football fans, if you have a crowd of, I don't know, Patriots and Giants fans going together, okay? And then you start pulling out Giants fans, only Giants fans. The proportion of New England fans will start to rise. Makes sense, right? But the size of the crowd will be smaller and smaller. And then you're pulling out New England fans. So you may end up with the same proportion, say half and half, but the size of the crowd will be really, really small because you pulled all those fans out. Same here, you're pulling out water, and then you're pulling out salts. You decrease the volume, and by pulling out a lot of salt, you actually decrease the concentration. Now, what's the role of Vasa recta? All that water and all that salt that leaves nephron loop. Think about water, actually. When water enters the medulla, what it should do to the medullary interstitial fluid. Yeah, decrease its osmolality. It should dilute it, right? In order to prevent it, Vasa recta picks up the water and takes it away. Now I'm going to ask you a hard question. So here's the thing. Look at the picture. This end of Vasa recta starts from efferent arteriole that leaves glomerulus. You with me so far? The volume of blood at this point is V1. As blood flows through the Vasa recta, on its way it picks up water and salts. You see what I'm trying to say? So water and sodium chloride, that leave nephron loop, they enter the blood in the Vasa recta. And the volume of the blood at the end, before it is discharged into vein, is V2. Which volume is bigger, V1 or V2? V2. Because blood picks up water and sodium chloride. So it goes from V1 plus blood and plus water and solutes to V2. Does that make sense? Do you understand what I'm saying? So the volume of blood that enters Vasa recta is lower than the volume of blood that leaves it. Am I clear? Does that make sense? We good?
Can you say that?
Sure. Of course. Now, blood enters Vasa recta, having volume V1. As it goes through Vasa recta, it is joined by the water and sodium chloride that just left the filtrate. So as it accumulates water and sodium chloride, its volume increases, and the volume V2 at the end of Vasa recta is bigger than the volume V1 at the beginning of it. Does that make sense? Cool. Now if we go back to the whole nephron story, we left the nephron at 100 milliosmoles in the distal convoluted tubule. So this 100 milliosmoles enter into the collecting duct and go deep into the medulla. Does that make sense? Now, that's 100. Look at the gradient of concentration in the medulla. It goes from 300 to 1200. Can water be reabsorbed in the collecting duct by osmosis? That's an easy question. Yes. The gradient perfectly facilitates it. Does that make sense? You understand that? The medullary interstitial fluid concentration is perfect. Can water directly cross cell membrane? No. Water is hydrophilic and the membrane is hydrophobic. Now this part here is facultative reabsorption. Now let me explain, hold on, facultative. Facultative is the opposite to obligate. Obligate must happen. Facultative, may or may not. Are we clear? Now, look at this scheme. Imagine that you are overhydrated. I'm going to avoid ADH by all costs so far, okay? Imagine that we are overhydrated. We drank like 2 gallons of water. What do we need to do with all that excessive water? No, God forbid, no. We drank like I'm standing here chugging like 2 gallons of water. What I'm going to do? I'm going to pee. I'm going to pee all that water. Does that make sense? Now, let's take a look at the regulation of the urine concentration. This reabsorption in the descending limb of the nephron loop, is it obligate or facultative? Obligate. Is there any regulation to it? No, I'm going to reabsorb that water anyway. Does that make sense? So I'm going to produce a whole bunch of diluted filtrate that will enter the collecting duct. Remember what I told you about the reabsorption in the collecting duct? I told you, can water be reabsorbed? And you're telling me, yes, it can. But will it be reabsorbed is the different question. If I want to pee it out, am I going to reabsorb it in the collecting duct? No. I'm just going to let this 100 milliosmol filtrate to leave my body in the form of diluted urine. Does that make sense? Now, different situation is I'm dehydrated. This part here in the descending limb, is this water going to be reabsorbed? No matter what, yes. And I'm going to produce fairly diluted filtrate. And this filtrate will enter collecting duct. This time I'm dehydrated. Do I want to reabsorb the water? Oh yeah, I do. So, collecting duct will start to reabsorb the water by now letting the water to cross the epithelial barrier. Are you with me? To cross the epithelial barrier and get in the medulla. You understand what I'm saying? We left out the key piece. Why? On the left picture, there is no reabsorption in the collecting duct. And on the right, there is. As Aaron mentioned, water cannot cross the membrane directly. What does it need to cross the membrane? A protein, a channel. Yeah, a channel. We call those channels aquaporins. So when you are very much dehydrated, remember, what's the dehydration? That's a simple and dumb question. What's dehydration? You lost water. When you lose water, what happens to osmolality, concentration of your plasma and extracellular fluid? It goes up. Okay? So that increased osmolality will be sensed by the chemoreceptors in your posterior pituitary gland. And your posterior pituitary gland will release the hormone called ADH, antidiuretic hormone. That hormone, ADH, will enter the bloodstream, get all the way to your kidneys, and will stimulate the synthesis of aquaporins in the epithelial cells of the collecting duct. The number of aquaporins will increase, water reabsorption will increase. How will it affect the volume of the urine? Small, concentrated. Does that make sense? You see how it regulates? Of course, you know, there's limit. You can't go without water for 2 weeks, hoping that ADH will take care of reabsorption, and you can't chug 4 gallons or 5 gallons of water, hoping that you're going to pee all of it out. Does that make sense? So, this is what happens when you overhydrated. Osmolality of your extracellular fluid or plasma goes down. You with me? Goes down. ADH synthesis is decreased and release is decreased. Number of aquaporins is decreased. There's no reabsorption in the collecting duct. You pee out a lot of diluted urine. If the amount of water, like you dehydrated, osmolality of the extracellular fluid goes up. Your posterior pituitary releases ADH. ADH stimulates aquaporin synthesis, and you have water reabsorption in the collecting duct. Does that make sense? We talked about it. I'm just curious if you remember. Why do you produce a lot of diluted urine when you drink alcohol? It inhibits something that we just talked about. ADH synthesis, yes. You produce less ADH, therefore you have less facultative water reabsorption. You produce a lot of diluted urine. Does that make sense? Basically, any ADH inhibitor will serve like a diuretic. Now, urea. How does it help? Okay, why do we need urea? So if you would take kidneys and you would look at the concentration of urea in the kidneys, you will see that it progressively increases as you descend deep in the medulla. So here's what happened. When the filtrate goes beyond that bottom tipping point, can you see the bottom tipping point? Here, the bottom tipping point. And the filtrate starts its travel through the ascending loop. Its concentration progressively decreases. So urea diffuses from the medulla into the filtrate. It's carried all the way to a collecting duct where, due to the water reabsorption, urea concentration increases dramatically. And then, as the filtrate moves down through the collecting duct, urea concentration in the filtrate always stays a little bit higher than the urea concentration in the medulla. So urea, as you can see, moves into the medulla. Does that make sense? And what's going to happen to that urea in the medulla? It's going to go back into the nephron loop. Essentially, you have a cycle. Urea cycles. Let me erase it and draw the cycle again. Urea cycles like this. Okay? Due to the differences in concentration, urea gets secreted in the medulla back into the filtrate, but it gets reabsorbed in the collecting duct to be secreted into the filtrate to be reabsorbed in the collecting duct. Does that make sense? And that movement of urea allows you to maintain the gradient. So high concentration, lower concentration in the medulla. That's the role of urea in the maintenance of the osmotic gradient. Okay, so really clinically important parameter is the evaluation of GFR. How do we evaluate kidney function? We measure the renal clearance. And the term renal clearance, the physiological sense of it, it's the volume of plasma that kidneys clear of that substance in a given time. Okay? Can be calculated using this formula right here, where U is the concentration of the substance in the urine, V, the flow rate, essentially GFR. Does that make sense? And P, the concentration of the substance in the plasma. We can use renal clearance in two instances. One, we can use it to measure GFR. Imagine that you have a substance. So say you give somebody this carbohydrate, okay? It's a polysaccharide. It's a plant polysaccharide called inulin, okay? And we know that inulin is completely filtered. What does that mean? It means that if concentration of inulin in the plasma is 1 milligram per milliliter. Does that make sense? Then the concentration of inulin, that's actually should be opposite. The numbers here a bit mixed up. Okay, numbers themselves. So if P is 1 milligram per mil and the U is 1 milligram per mil, make sense? Then, and the GFR, normal GFR, is 125 mils per minute. Then renal clearance for inulin is going to be 125 milliliters per minute. So, how we use this parameter, renal clearance, to measure GFR? We experimentally know that certain human metabolites are freely filtered. For instance, creatinine. Have you heard that term? creatinine. It's the product of creatine phosphate breakdown. It's freely filtered, meaning that all creatinine that is in your plasma will end up in the filtrate. That makes sense? Do you understand what I'm saying? So for the substance that is freely filtered, the renal clearance is equivalent to GFR. I'm going to let it sink for a second. Forget about the kidneys. Forget about the kidneys. Okay? Forget. Do you have filters at home? Do you have filters at home? Imagine that you pour some water with a dye on the filter. Like 1 pint of water that has a certain amount of dye added. Right? You wait for all this water to filter through, and then what you see is that your filtrate has the same color, contains the same amount of dye. That makes sense? This means that dye is freely filtered. We're clear? It means that concentration of the dye in your filtrate doesn't really change. Am I clear? So same happens with the measurement of GFR. If the concentration of compound in the blood and the concentration of compound in the urine are the same, it means that this compound is freely filtered. It means that its renal clearance is equivalent to GFR. It means that every minute 125 milliliters of your plasma are cleared of that compound. Why it's important? When you go to do a blood work and the urinalysis to your physician, they will take a sample of the urine, and they will take the sample of the blood, and they will measure creatinine. For creatinine, renal clearance is equivalent to GFR. If the concentration of creatinine in your blood and the concentration of creatinine in your urine are the same, you're fine. Your kidneys are fine. Does that make sense? If the filtration membrane is damaged, you may have more creatinine in the urine, less creatinine in the urine. It all can change. Does that make sense? So creatinine is a really convenient way to estimate GFR. Basically, if your filtration membrane is damaged, then the concentration of the plasma creatinine will be lower. And your GFR will show up not as 125, but says 90, or 70, or 60. That make sense? And that will be a signal for the chronic kidney disease. There are clinical criteria, which I don't remember, to be honest. I believe that 125 to 90 GFR is the pre-disease state. Below 90 is chronic kidney disease. Below 60, I think it's below 60. I'm not sure if it's 60 or 40. It's a kidney failure that requires dialysis or kidney transplant. Does that make sense? Now, that's use of renal clearance to measure GFR. Now, how does renal clearance relate to the behavior of the substance in the kidneys? As we mentioned, if renal clearance is 125, the substance is freely filtered. Can you see that? Do you understand what I'm saying? There's no reabsorption, there's no secretion. It just goes through the glomerular filtration membrane. Okay, it goes from the blood into the filtrate. Are you with me on that? Do you understand what I'm saying? If renal clearance, think about the name, renal clearance is 0. What does that mean? Is any plasma cleared if it's 0? If clearance is 0, any plasma, no, plasma is not cleared of that compound. It means that compound is completely reabsorbed. Example of the compound with a renal clearance of 0 is glucose. In the normal healthy kidney, glucose is completely reabsorbed. Does that make sense? If the renal clearance is between the 0 and 125, compound is partially reabsorbed. Okay, so it's filtered first and then some of it returns back into the blood. And finally, if renal clearance is more than 125, the compound is secreted. Say, for penicillin, as far as I remember, renal clearance is more than 125. So the concentration of penicillin in the urine will be more than in the plasma. You will be filtering it and then actively secreting. Does that make sense? So this is something I'm going to ask you on the exam. I guarantee that. So I'm going to give you a compound and I will tell you renal clearance for that compound is 60, which is less than GFR. What does that mean? Compound is partially reabsorbed. Am I clear?
So 125 is the
125 is the GFR, and when I ask you a question, any question on any exam or quiz, unless I specify that it's a disease, I presume that we're talking about the normal physiology, healthy physiology. Any questions? You good? Now we're going to talk about the balance of certain key chemicals. We're going to start with water. So this is where you get water from. 10% of your water is produced by your metabolic processes, your cellular respiration. You take a certain amount with food and certain amount with drinks. So on this image, drinks account for 1.5 liters. Have you heard about the recommendation of drinking like 4 liters of water a day? There's no clinical evidence to any benefits from it. None. I mean, if you live in Louisiana, it just keeps coming back. But if you live in Louisiana and you work outside for 12 hours a day, then yeah, you're going to be drinking like way more than 1.5 liters, but that's because you lose water. If you're not sweating profusely, that's the amount you should be drinking every day. Okay? Excess water will be excreted. Now, how you lose water? Look at this. You lose water in feces. That's not regulated. Well, a little bit. You lose water in sweat. And you lose water by evaporation, mainly through the lungs. When you exhale some water vapors. The main regulated part of water conservation comes with urine. We're good? Now, let me ask you this very, very quickly. What is more of a problem? Overhydration or dehydration? Dehydration. I mean, unless you drink 6 gallons of water, as one woman in California did. They had a competition of who can drink more water. She won. She died next day because of the brain edema. But she won. So, unless you do this, you know, if you drink a little bit more than usual, like you drink, 3 cups of tea instead of 1 cup of tea, you're going to just excrete all the extra water. Dehydration becomes a problem. So what happens when you are dehydrated? First and foremost, the volume of plasma goes down, and the osmolality of extracellular fluid goes up. these two consequences of dehydration, do they make sense to you? They do? It's perfect. They should. So there are several mechanisms responding to these changes. First, when extracellular fluid osmolality goes up, that stimulates osmoreceptors, the chemical receptors in the hypothalamus. You feel thirst. When you're thirsty, what do? You drink. There's going to be a lot of really stupid questions down the road. Okay, you drink. Problem solved. Correct? Now, when plasma volume goes down, your blood pressure goes down, which stimulates granular or juxtamedullary cells in kidneys. They release renin. Renin activates angiotensin. Angiotensin stimulates thirst. That makes sense? What else does angiotensin stimulate? Which hormone? So renin, angiotensin, and the third, aldosterone system. So your aldosterone will go up, which is not here in this example, but your aldosterone will go up. And aldosterone will increase the retention of what? Sodium. And retention of sodium will increase the retention of what? Water. Got it? And on top of that, elevated osmolality and decreased blood pressure will stimulate the release of antidiuretic hormone. Antidiuretic hormone will increase facultative reabsorption of water in the kidneys, normal osmolality of extracellular fluid, normal pressure, but low volume of concentrated urine. Clear? Good? Any questions about this? So these are mechanisms of water balance regulation that you need to know by ADH, via hypothalamic thirst center, and via renin, angiotensin, aldosterone system. Good? Sodium. The sodium concentration is regulated extremely well. What you actually may change is the sodium content in the body. Okay? Now, here's the thing. Imagine that you have low blood pressure, which corresponds to the lower sodium concentration in the blood. So basically water starts to leave the blood, lower sodium content. Basically decreased sodium will increase the release of renin, okay? And that will stimulate adrenal cortex and stimulate aldosterone. Actually, I can give you a more detailed mechanism. Just think about this. Remember we talked about juxtamedullary mechanism of regulating GFR. When your blood pressure goes down, your blood flow, your filtrate flow goes down as well. Do you understand what I'm saying? So your blood pressure goes down, blood flow through the kidneys decreases, amount of filtrate decreases, filtrate flow decreases, sodium is reabsorbed more efficiently. Okay? What happens to the sodium concentration in the filtrate? It goes down. That signals to granular cells to release renin. Renin will stimulate angiotensin. Angiotensin will stimulate aldosterone. What you're going to be retaining? Sodium and then water. Sodium and then water, yeah. You will reduce you will restore the sodium concentration. Does that make sense? The side effect of aldosterone release, secretion of potassium. But we'll get back to this. Okay? Does that make sense? Now, if your blood pressure... Now, here's the absolutely marvelous thing. How sodium concentration is actually regulated. Let's play this whole story alone. Ever heard about famous KFC sandwich in which 2 buns were substituted with fried chicken? Ever heard about it? What made it famous, except for sheer awesomeness of that idea, I think it's 150% American as it can be. It's the amount of sodium that one sandwich contained. One sandwich contained 2 daily doses of sodium that the person should eat. So imagine that you eat, your daily norm is about 2.3 grams. You eat 4.6 in one sandwich. I'm not counting other foods. So what happens to the sodium concentration in your extracellular fluid shortly after? It skyrockets, right? But sodium concentration means osmolality. Osmolality's got to be controlled. So how can this be controlled? What do you need to do with the concentrated sodium? Drink water. You're going to feel thirsty, but it takes time to drink water. You have to dilute it. So your extracellular fluid will be pouring water from your intracellular fluid, from your cells. Okay? Now, when this plasma sodium gets diluted by adding water, what happens to the volume of blood? It increases. What's going to happen to your blood pressure? It increases. Actually, a colleague of mine, who's quite ironically is the director of Cardiovascular Research Institute down in Louisiana, he tried this sandwich. He said that he felt his blood pressure skyrocketing. It was an inhumane experiment on himself. Okay, but you feel it going up. Okay? Now, when your blood pressure increases, the hormone called atrial natriuretic peptide is released from the atria of your heart. Okay? Now that leads to inhibition of ADH release, inhibition of aldosterone release, and inhibition of angiotensin II release. So angiotensin, look, can you see that? So ADH and aldosterone both can regulate reabsorption of water and sodium. So that skyrocketing blood pressure will reduce sodium reabsorption. That makes perfect sense. You just ate 2 daily doses of sodium, right? So instead of reabsorbing sodium, you will start to excrete it. And it will lead to the decreased reabsorption of water. So you will start to pee out quite a lot of salty urine. You will reduce the water volume and you will reduce the sodium amount. Does that make sense? This is the reason the American Heart Association recommends to limit sodium to the people with high blood pressure. Because when you eat salty food, you draw water into your extracellular fluid, you elevate your blood pressure constantly, you're at the higher risk of, hypertension associated damage. That makes sense? And consumption, constant consumption of salty foods will inevitably lead to hypertension. Does that make sense? In terms of angiotensin, angiotensin is a pretty powerful vasoconstrictor. When its levels go down, vasoconstriction goes down, blood pressure goes normal. So basically what I'm trying to tell you, to convey to you, Sodium concentration is pretty tightly regulated, and it will closely tied to the blood pressure. Basically, if you eat a lot of sodium, your blood pressure is going to skyrocket. Does that make sense? Calcium. That's the simplest, that's a sweet and simple story. Low calcium, parathyroid hormone, increased calcium reabsorption in kidneys. Is that understood? Do you see that? That's it. Like, I'm not asking for any more details. All you need to know about calcium reabsorption in kidneys, regulated by parathyroid hormone. You clear? Now, potassium. Did we talk about potassium? Yeah, we did talk about potassium and capital punishment. Okay, so potassium is really tightly regulated. Okay, let me explain what happens in the potassium case. So, when you eat something with a lot of potassium, the plasma concentration of potassium goes up. Does that make sense? And then potassium is excreted by two ways. One, direct excretion. It's filtered out. And second is aldosterone mediated. Let me explain what I mean. It's going to be very schematic. I wouldn't take what I'm going to draw now as the exact representation of like physiological scale and numbers, but it gives you a pretty good understanding. So on the x-axis is potassium in plasma, on the y-axis is potassium in filtrate. Does that make sense? So the blue line reflects a direct secretion of potassium. Does that make sense? You eat more potassium, it is directly filtered and excreted by kidneys. No aldosterone involved. Does that make sense? Now the green line is going to reflect the concentration of aldosterone. So you see, the more potassium you have in the plasma, the more aldosterone you're going to produce. Does that make sense? And finally, the red line reflects the total potassium excretion. Now compare the red and the blue. Blue is the direct excretion, no aldosterone involved. Red is total. That difference, where does it come from? What's responsible for that difference? So that total excretion of potassium consists of direct one and the one that's regulated by which hormone? Aldosterone. So you have two ways. Does that make sense? The direct excretion and aldosterone mediated. We're clear? Break, quiz review, and then acid-base regulation.